Hello everyone, welcome to my channel Botany for Neet. Let us solve a question asked in Neet examination. Unequivocal proof that DNA is the genetic material was first proposed by. What is the meaning of unequivocal? That means a proof which was finally guaranteed with no ambiguity, no confusion at all. Okay, who first gave that proof with the now it was completely accepted by all scientists without any doubt that DNA is the genetic material. It was first proposed by whom? Griffith, Frederick Griffith, Alfred Hershey and Martha Chase, Avery, McLeod and McCarthy, Wilkins and Franklin. Okay, before we understand, let us see what were the experiments which were done by Griffith, Hershey and Chase, Avery, McLeod, McCarthy, Wilkins and Franklin. Now, nuclein or the nucleic acid was first discovered by Frederick Measure. He first discovered nucleic acid, but what actually, which biomolecule? Is it DNA or protein or is it the RNA, ribonucleic acid, which acts as genetic material? Genetic material means which is actually getting transferred from mother to the progeny. Out of these three, which actually is responsible for acting as a genetic material to pass on the traits present in the mother to the progeny? So that was the question among the scientists. So they have done various experiments, and the question here is who actually finally proved through an experiment that it was DNA, that is the genetic material or that acts as genetic material, which helps in passing on the traits from parent to progeny. Okay, so before understanding all experiments, I would like to tell you about Petri plate. Now see, when I have to grow a plant, we know we take a pot, inside the pot we put soil and put either I sow the seeds or put the sapling, then pour some water and keep it in sufficient amount of sunlight, right? But what about microbes? If I want to grow microorganisms, then what should I do? Where will I grow them? We have Petri plates. Okay, these Petri plates can be either of glass or they may be plastic. Okay, and you can see this is the lid of the Petri plate. And just like how plant when you sow a seed or plant, we first put soil in it which is rich in nutrients. In the same way here, if you directly put bacteria in it, will it be able to grow even if I give temperature, sunlight, everything? No, obviously no, it requires nutrients. So we pour media in it. Okay, basically media is nothing but nutrients. We give nutrients to the microorganism. Okay, and agar is added. It acts as a solidifying agent on which this microbes can grow basically. Okay, so Petri plate is basically where you can culture microorganisms. That means we can grow microorganisms. Okay. Okay, now let's dive into the experiments which were done by various scientists. First is Griffith's experiment. What he has done, he has taken a specific type of bacterium, Streptococcus pneumonia. It is also known as pneumococcus. Okay, the Streptococcus pneumonia is also called as pneumococcus, and it causes pneumonia disease. Okay, this Streptococcus, what he has done is he has taken this Streptococcus and grown them on Petri plate. That means what? He has taken Petri plate, put some media onto the media. Now he had added an inoculum. That means some few cells of Streptococcus pneumonia. Streptococcus pneumonia, there are two strains available. On what basis are we saying that there are two strains available? That means when you allow the cells of the streptococcus to grow on a petri plate, they form either rough colonies or smooth colonies. Okay, rough colonies, that means the texture of the colonies when you see on the petri plate, when you see the cells on the petri plate, they're slightly rough in texture, while the smooth colonies are quite smooth. So I'm sorry, R2 strain produces rough colonies, S3 strain produces smooth colonies. Why is it so that S3 strain is producing smooth colonies? Because it secretes a polysaccharide coat. And this polysaccharide coat is also giving it virulence character. That means ability to infect and cause death of host cells. Okay, so what actually Griffith has done is he took Streptococcus pneumonia, also known as pneumococcus, grown them on a media where there are two types of strains which were available, R2 strain and S3 strain. R2 strain is a virulent strain, that means it does not cause any infection, means there is no way it can cause the pneumonia disease. Okay, while S3 strain causes pneumonia disease as it can produce polysaccharide coat. Now, what is Griffith's experiment? He has actually done? He took this S3 strain, living S3 strain, that means live Streptococcus pneumonia S3 strain was taken and injected them into mice, basically your rats. He took the S3 strain and injected into mice. We know S3 is virulent, it has polysaccharide coat, it can cause pneumonia. So mouse the mice, they died because of pneumonia disease. Okay, as expected only, right? He has taken the virulent strain, that is Streptococcus pneumonia S3 strain, injected into mice, and the mice died because of this pneumonia disease. Now he has taken live R2 strain. R2 strain, it is avirulent. Okay, so he has injected them into mice. What is the result that we expect? Obviously, the mice will not die. Avirulent strain, so no pneumonia here. Okay. Now, what he has done is he has taken this S3 strain of pneumonia and exposed them to very high temperature. As a result, what happened? All the bacteria got dead. See, Streptococcus pneumonia is a type of bacterium. So the bacterium, it died. Now this heat killed S3 strain. That means the dead Streptococcus pneumonia cells he injected into mice. And what happens? Obviously, when the bacteria is dead, can it cause any infection to the host cell, which is mice here? Can it cause any infection? Obviously no. So the mouse lives. So in all these three cases, case 1, case 2, case 3, the results were as expected. In the first case, he took a virulent strain. Virulent strain means a strain which has the ability to cause infection. S3 strain he has taken, injected into mice, and the mice died. In the second case, he has taken a virulent strain, injected into mice, and it did not die. In the third case, he has taken S3 strain, which is virulent strain, heated it, as a result of which the bacterial cells died. These dead cells he injected into the mice. Then what happened? The Obviously the mice were not killed. Now in the case 4, there was an interesting observation that he has made in case 4. What was it? Let's see. He has taken heat killed S3 strain and mixed them with R2 cells, live R2 cells. Living R2 cells, when they are mixed with heat killed S3 strain and injected into mice, what would we expect? We expect that mice or mouse will be alive. But to his surprise, what he found was the mice was dead. And indeed, when he tried to extract the cells from these mice, he found that living S3 cells were recovered. That means what? From this experiment, he analyzed and hypothesized that there is something which is present in this heat killed S3 strain that is getting transferred into R2 strain, as a result of which R2 strain is getting converted. It is now having the ability to produce that polysaccharide coat and cause virulence in the mice. So some material is getting transferred from S3 strain into R2 strain, that is changing the R2 strain into S3 strain, as a result of which he could recover live S3 cells from the rat, the dead mice. Okay, and this change from R2 strain into S3 strain, he called it as transformation. There was a change from the R2 strain to S3 strain that he has recovered from the cells of the mice. But he couldn't actually explain chemically what this transforming principle was. That means, is it RNA that is getting transferred from S3 strain into R2 strain and causing that virulence nature, or is it the DNA, or is it protein? He didn't explain all that. But through his experiment, we found out that there is a transforming principle, or there is a substance that is getting transferred from the heat killed S3 strain into R2 strain, which changed the R2 strain into virulent S3 strain, as a result of which the mouse died and he could recover living S3 cells. So through this experiment, they could just like Griffith, he worked on streptococcus pneumonia, which causes pneumonia disease in any host cell or the bacterium, whatever host it affects, it causes pneumonia. And streptococcus pneumonia is also called as pneumococcus. And through this experiment, they try to tell that there was some factor, some substance which is changing this S3 strain, sorry, R2 strain, the live R2 strain into S3 strain, and these cells were recovered from the mice. This was the interesting case study that he had uh, found out through his experiment. But are we now clear through this experiment that which is the genetic material? Obviously, no. We still don't have idea out of RNA, DNA, and proteins, which is the genetic material. Now, three scientists, Avery, McLeod, and McCarthy, they have done another experiment to show that it is indeed DNA which is the genetic material and not RNA or proteins. It is indeed DNA which acts as genetic material. How did they prove? What did they do? They took heat killed S cells. That means heat killed S2 strain, S3 strain cells they have taken. Okay. Now we know that bacteria also has sugars in it, lipids in it, RNA in it, right? So what they have done is they have taken the cells, bacterial cells, the suspension of the bacterium, from where did like the heat killed S cells. So they have taken the bacterial suspension. This bacterial suspension, they have treated it with three different enzymes. Usually enzymes end with the suffix ase. Proteinase is the enzyme which breaks down proteins into its constitutive amino acids. Okay. In the same way, RNase is the enzyme that breaks down RNA into nucleotides. In the same way, DNase is the enzyme that breaks down DNA into deoxyribonucleotides. Is it clear? So, what he has done is he has taken the bacterial suspension and treated it with proteinase. Then, what happens? All the proteins get killed. Whatever proteins are present inside the strain, heat killed S3 strain, all are uh, not killed, denatured. That means the proteins are broken down into amino acids. If protein was the transforming principle, then what would have happened? Since there were no proteins, but still they could observe that when R cells, that means R2 strain cells, were added into the beaker, they could recover S3 cells. Though proteins were completely denatured by adding proteinase, still there is the transforming principle present, that is converting this S3 cell, the heat killed S3 cells which were present. First, he has taken heat killed S3 cells, then to it they added R2 cells. These R2 cells, which were present, the live R2 cells, they got converted into S3 cells, living S3 cells. If protein was the transforming principle, we would have not seen this. But even after adding proteinase, still you can observe that S3 cells were recovered. That means protein is not the genetic material. So protein is ruled out. So now our doubt is it can be either RNA or DNA. So what they have done is they have again taken the heat killed S3 cells. To this they added RNase. RNase is the enzyme which attacks RNA and completely breaks it down. Now what happened? Then they added R2 cells, live R2 cells to it. Again they observed that R2 cells are getting converted into S3 cells. That means what? RNA is also not the genetic material because RNA is completely broken down by RNase enzyme. And we have already ruled out it's not proteins. So then what they did is they have taken S3 cells, that, that means heat killed S3 cells, added DNAs to it. What does DNA do? It breaks down, DNA's enzyme do, it breaks down the DNA. This time, what they have observed is when they added R cells to it, there were no S3 cells that were retrieved. They did not get S3 cells. That means what's happening? R2 cells, the live R2 cells, were not getting converted into S3 cells. Why? Because there is no DNA. So this DNA which is heat killed in the S3 cells, the DNA which is present, it was completely broken down by DNase enzyme. As a result, There is no way R2 cells were able to synthesize the polysaccharide coat. Therefore, they could not recover S3 cells. This experiment proved that DNA was the genetic material. It is actually the transforming principle. They found out that it is indeed the DNA present in heat killed S3 cells that is getting transferred into R2 cells, synthesizing the polysaccharide coat and converting into S3 strain. That's the only reason they could recover S3 cells out of the dead mice. Every McLeod and McCarthy, they proved that it was not proteins or the RNA, but it is indeed DNA which is acting as the genetic material. But many of the scientists, they didn't accept, or they were not completely uh, accepting the hypothesis or the findings of every McLeod and McCarthy. Okay, later then Hershey and Chase, they have done a remarkable another experiment where they worked with bacteriophages. Okay, they didn't work with bacteria like Griffith or every McLeod McCarthy. Every McLeod McCarthy and Griffith, they worked on Streptococcus pneumonia, R3 strain and sorry S3 strain and R2 strain. But Hershey and Chase, they worked on bacteriophages. What basically are bacteriophages? These are the bacteria which are infected by viruses. They are called bacteriophages. Okay, and they worked on T2 bacteriophage. Okay, these bacteriophages, and they were infect they what are bacteriophages? I told these are the viruses which are infecting bacteria. They worked on E. coli, Escherichia coli bacteria. Okay, you can see here, this is a bacterium cell. Consider this is E. coli cell which is being infected by a viral particle. Okay, now what actually they have done is they have grown few bacteriophages in a petri plate containing medium with radioactive labeled or radio labeled phosphorus 32. Okay, and few bacteriophages, they have labeled radio labeled them with S, sulfur. Okay, phosphorus and sulfur, radio labeled phosphorus and radio labeled sulfur. So what they have done is they have taken this bacteriophages, allowed them to grow in a petri plate which was radio labeled with sulfur and radio labeled with phosphorus. Now, let us understand what was the experiment that they have done. Okay, see, this is the bacterial cell. Initially, what actually happens is this is your bacterial cell, E. coli cell. When it is infected by virus, first virus, what it does is it fixes, it adheres, it gets attached itself to the membrane of the bacterial cell. After adhering, what it does is the nucleic acid which is present inside the viral particle, it slowly injects it inside the bacterial cell. Now, once the viral genetic material enters into the bacterial cell, the bacteria thinks that it is now the bacteria starts synthesizing the proteins which are required for the virus. It thinks that it is its own genetic material and starts synthesizing the proteins which are required for the virus. Okay, so that is the mode how the virus actually attacks the bacteria. Now let us understand what was the experiment which was conducted by Hershey and Chase. They have taken bacteriophages. Now here you can see the viral protein coat. V- See, viruses, they have protein coat and DNA to them. Okay, DNA, it is rich with phosphorus. Okay, and protein coat, it is rich in sulfur. Okay, so what they have done is they have grown these bacteriophages in a medium containing, in this case, they have grown the bacteriophages in a medium which is radioactively labeled with sulfur. When it is radio labeled with sulfur, Su- then will the viral protein coat will be radio labeled or viral DNA be radio labeled? Obviously, viral protein coat is radio labeled because sulfur is radio labeled. So that's the reason viral protein coat is radio labeled. In other case, they have grown the bacteriophages in a medium containing radio labeled phosphorus. So radio labeled phosphorus binds to DNA or to the protein coat, obviously to the DNA. Now, when they allowed these bacteriophages to grow in the medium, what happened is, we know that once the virus attaches to the bacterium, it injects its DNA into the bacterial cell. So, what is infection? Infection means the virus getting attached to the bacterial cell, and slowly it will release its genetic material into the bacterial cell. Then, by blending, what we do is we separate the bacterial cell from the viral ghost. Okay, this is called ghost. Without any DNA material, the viral coat protein which is present, we are separating the viral protein coat from the bacterial cell, where the bacterial cell is carrying now the viral genetic material. After blending, we do centrifugation. Why we do centrifugation? To separate the protein coat from the bacterial cells. Okay. When they separated the protein coat from the viral bacterial cell, the protein coats were present in the pellet, and the bacterial cell were present in the supernatant. What do you understand by pellet and supernatant, for example? Consider a tube. I have added water and sand. After some time, what happens? Does sand really dissolve in water? No. Obviously, you get two layers. One is the sand layer, and the other layer is water layer. This is called the pellet, the down part, and the above part is called supernatant. In the same way, what they have done is allowed the viral particle to infect the host cell. Then genetic material is injected inside the bacterial cell. Then by blending process, we separate the bacterial cell and the viral protein coat. Now by centrifugation process, what happens is there will be a pellet form and supernatant form. What they observed is, if we consider that protein is actually the genetic material, let us consider protein is genetic material, then what should happen? You can see that here the protein is radio labeled with sulfur. If protein is the genetic material, then I should observe that the bacterial cells should show me the radio labeled sulfur, right, in the pellet. But the pellet which is formed here was not radio labeled. But in this case, where the DNA is getting transferred into the bacterial cell, here what happened is DNA is showing the radio labeling of phosphorus. So protein code has not entered into the bacterial cell, because protein is not the genetic material. If protein would have been the genetic material, it would have entered into the bacterial cell and showed me the radio labeling. But here what happened? Protein is not the genetic material, it is not entering into the bacterial cell. So no radio labeling seen in the cells. But here what's happening? DNA is the genetic material, we know, so it is entering inside the bacterial cell and it is showing me radio labeling. This, indeed, this experiment which was done by Hershey and Chase, it proved without any doubt, it gave an evidence that it is indeed the DNA which is acting as the genetic material, okay, and not protein. Why is it so? Because if protein is the genetic material, then what would have happened? See here, protein code is radio labeled with sulfur. If, if protein is genetic material, it would have entered inside the bacterial cell and showed me radio labeling in pellet. But there was no radio labeling seen in pellet, no radio labeling seen in the supernatant as well. Why is it so? That's because it's not entering inside the bacterial cell. Okay, DNA is entering inside the bacterial cell, so it is showing the radio labeling. You can see in the supernatant. This proved that DNA is the genetic material. So let us see the question now. Unequivocal proof that DNA is the genetic material was first proposed by Alfred Hershey and Martha Chase. Griffith conducted the experiment, but he didn't tell which is the genetic material. He just said that there is some material which is getting transferred from the heat killed S3 strain into R2 strain. It is getting transferred and converting this R2 strain into S3 strain. And every McClure and McCarthy, they have conducted experiment and showed that DNA is genetic material, but scientists did not completely accept it. Later, Hershey and Chase, they have done experiment to unequivocally prove that it is the DNA which is the genetic material by working on bacteriophages, by taking radio-labeled phosphorus and radio-labeled sulfur, S35 and P32. Okay, Wilkin and Franklin, they have done X-ray chromatographic studies which helped to get DNA structure. Okay, like Watson and Crick, they have given the structure of DNA that it is double helical. Yes or no? Watson and Crick, they first gave the complete BDNA structure. Their structure was based on findings of Wilkin and Franklin's X-ray crystallographic studies only. Okay, so the correct option is option two. I hope you understood the solution for this question. If there are any doubts, please feel free to comment down below. Thank you. Have a good day.